making nutrients like lactose? How about getting rid of drugs, efflux pumps? Efflux pumps pumping out toxic material outside of the cell. Okay, so here is sort of in text form. And we'll look at this in the diagram. So, understand in chapter 13, though I didn't cover it in detail, understand what glycolysis and TCA cycle is about. It is glycolysis. It's the lysis of glucose into two molecules of pyruvate. And why does the cell do that? It does it to make ATP and to make NADH. The TCA cycle takes the pyruvate and then completely oxidizes it to CO2. So that by the end of the TCA cycle, you have completely oxidized glucose to carbon dioxide. You have stripped every electron available, biochemically available, from glucose by the end of glycolysis and TCA cycle. So, understand that's what we did in chapter 13. Now we answer the question, how are those electrons being used to make ATP? So they're being carried by NADH or FADH2. There's two main biochemicals that are electron carriers. And so those electrons then go to the membrane, whether it's the plasma membrane in bacteria or in our cells, the mitochondrial membrane in the organelle of the mitochondria. Same process happens. So this is where the electrons are donated to this FMN protein, which really, I should be consistent and call it the NADH dehydrogenase. FMN is a component of the NADH dehydrogenase protein. So I make that correction in your figure. It's not exactly wrong, but it's more clear if I keep it consistent. And we'll look at the details in the next slide. So with a flow of electricity, with electron transport in those membrane proteins, protons are pumped outside of the membrane, and more protons then are on the outside than on the inside, and this generates this proton motive force, which is this primal energy source, which results from electron flow. So electron flow first, proton motive force second, ATP production finally third. So that's why I say electron flow is the primal energy source. I say proton motive force is the primal energy source, but understand it is as a result of electron flow. Okay, here's a really nice figure in your diagram. These big blobs in the membranes are getting more and more realistic as we get X-ray crystallography information from these massive proteins. And so the protein that we're talking about here is this NADH1 complex. And it actually sticks out into the cytosol, into the cytoplasm of the cell, and then connects to this series of proteins that are in the membrane. So there's something like, it's a multi-dimensional, at least 14 subunits. The NDH, the NADH dehydrogenase enzyme that we have in our mitochondria has like 56 subunits. It's all the more massive and complex. But it serves the same function, and that is, let's just, NADH donates 2 electrons. Now, where did NADH get those electrons? From glycolysis, from TCA cycle. So, connect chapter 13 to chapter 14. All of glucose's electrons are now being carried by NADH or FADH2. And so here is a pair of electrons, which is what NADH carries. So when NADH donates those electrons to the NDH protein, NDH NADH becomes oxidized, NAD+. It has lost those electrons. So those electrons are passed from, there's a few metal cofactors. There's iron in these proteins, and they are transferred through this massive membrane complex to ultimately, well, the first other enzyme involved is this coenzyme Q, which becomes reduced. And you know it's reduced here because the electrons are right here, QH2. You can follow the electrons by identifying where the hydrogens are. And so here's that pair of electrons. So as a result of this electron flow through this NDH complex, look, protons are pumped from the cytoplasm to the outside of the cell. So there's this first big installment of protons being pumped and the beginning of this proton motive force chemical and charge gradient. And it's a result of what? Electron flow. The flow of electricity. And we can call it that because when we tap into it, we can run light bulbs, or we can run devices. So, anything more I can tell you about this? Huge enzyme complex, transfer of electrons that were from NADH. It's a pair of electrons. One pair of electrons through this complex allows for the pumping of about 4 protons. And then the first electron acceptor downstream of NDH is coenzyme Q. So this is the beginning of the electron transport chain. But hey, it's important because it transfers electrons from the cytoplasm now into this membrane-bound protein complex. Okay, so this is just what we've looked at here. Kind of in the older fashioned, more simpler form. But you know, we really we're just a much better understanding. The protein chemists have given us this data, and we've got all these hydrophobic alpha helices here, which is where these protons are being pumped. And so a little bit to review this again, RH2 is basically some sort of reduced compound. I would prefer just, hey, where did NADH come from? It came from the TCA cycle in glycolysis, okay? So there's the, recapping what we just covered, 2 electrons being transferred and now being reduced, going to reduce coenzyme Q, and here it is, coenzyme Q is now reduced. Now, those electrons are just transferred from other coenzyme Qs in the membrane, and then they ultimately go to the cytochrome B oxidase enzyme. That's what this is shown here. And tracking those electrons, they go to reduce oxygen. So oxygen is, we're not quite ready for that yet. Oxygen is the final electron acceptor for those, for that pair of electrons. This is aerobic respiration. And you know, I said in chapter 13, there's 3 main stages: glycolysis, TCA cycle, respiration. We are looking at respiration now. Okay? Where does it get that word respiration? Oxygen is the final electron acceptor. That is the fate of that pair of electrons, is they go to reduce oxygen, and the reduced product of oxygen is water. Okay? So, basically 3 protein or 3 biochemical components to the electron transport chain. This is a little bit simplified, but it represents a lot of electron transport in a lot of organisms. NDH1 transfers electrons to this coenzyme Q pool, which ultimately transfer to cytochrome B oxidase. and those electrons are transferred to oxygen to be reduced and to form water. Okay? Not too complicated. So in the meantime, more protons are being pumped. So we already talked about the four protons being pumped from NDH. Other protons are being pumped as a result of electron flow. So another 4 protons have been pumped, typically from that single pair of electrons. So all of these protons, and I went ahead and added this part, is, well, what happens to those protons? They go right into, because ATP synthase is right in this membrane, right next to NDH and the coenzyme Q pool. And so I just want to make sure this is not some separate cell or separate compartment. The ATP synthase enzyme complex is right in that membrane, and those protons right here, you can barely see them on this chart, but you should have this complete figure, are then used by this incredible biomotor to synthesize ATP. And we'll look at that in some detail. But I want you to then see the complete story from glycolysis, step number 1, TCA cycle, step number 2, and this is respiration, including the use of these protons to make ATP. And there's the whole metabolism right there on this chart, right here on this figure. So before we go on and talk about ATP synthase, any question about maybe this big picture view of the 3 steps of metabolism: glycolysis, TCA cycle, and respiration?
Is the TCA the Krebs cycle?
Yes, good question. The TCA is the Krebs cycle. So Dr. Krebs many years ago made the discovery, and we call it the tricarboxylic acid cycle, because many of the intermediates of the TCA are tricarboxylic acids. Yes, TCA equals Krebs cycle. Any other questions? Okay. Now, ETS animation. I thought I was only going to show the animation of the ATPase. We'll see. I probably will also show this. It's helpful. But let me review ATP synthase. Okay, let us see. In the big picture, you've got F0 and F1, the F0 complex and the F1 complex. So it's a massive multi-subunit enzyme, but it's got 2 main subunits, the F0, which is embedded in this membrane, and then the F1, which is then aimed to the inside of the cell and is in the cytoplasm of the cell. So remember, always orient yourself when you see a membrane, what's the inside, and what's the outside and what's the inside. So F0 embedded in the membrane, F1 protruding into the cytoplasm. Highly conserved protein complex, so this is the very, very similar structure to our ATP synthase, as in bacterial, as in fungal, as in plant ATP synthase. Okay, so one of the key components are protons. So we've already said we're generating this massive, there's 100 times more protons on the outside of the cell than on the inside of the cell because of electron flow causing protons to be pumped. What does the cell do with the protons? Well, this subunit A is actually a proton channel, because realize this membrane is highly impermeable to protons. They will leak through slowly, but generally, if there is not this proton channel in ATP synthase, they remain on the outside. And so this component of ATP synthase right here is a hydrogen ion channel, And so that's then where the transport happens. And so proton flow causes the C subunit. And so in the F0 subunit are these series, and there's like, I don't know how many of them. There's quite a few of these. There's 12. How about I say 12 subunits of C. And the proton flow right through this port causes these subunits to rotate. And there's an axle. You know, you're going to see the mechanics of this because it works just like a cam in an offset cam in a motor. So you get rotation. Proton flow causes this set of subunits to rotate around the axle, and then the rotation causes the F1 subunit to phosphorylate ADP to ATP. And we're going to look at a little bit more detail as to how that happens, especially in the movie, which shows you some of the motion. Okay, so in a little bit more cartoon fashion, here is the flow of protons through the A subunit. And here you can see a little bit more clearly the 12 C subunits, which are surrounding this axle that connects the F0 membrane subunit to the F1 cytoplasmic subunit of ATP synthase. And proton flowing through that proton channel causes this, the protons accumulate temporarily around the C subunits and cause this rotation. So in the end, you are having a spinning ATP synthase, including this axle that transmits the energy to the F1 subunit. Now, the only thing I want to point out here in this part of the subunit is this is not a perfectly round axle. It's not a perfectly round shaft. It is what we would call in mechanics a camshaft. So it has a cam to it, and so when this thing rotates, it causes this entire subunit to bulge and to bulge out. And it is that bulging out that is then driving the synthesis of the ATP, the phosphorylation of ADP to ATP. And I think when we watch that movie, it'll be a little bit more clear. But it becomes a biophysical motor. So these are physical. It's interesting to see chemistry, proton flow chemistry, converted to physics, meaning this camshaft is rotating, and then turned back into chemistry, the phosphorylation of ADP to ATP. So it's just to say biophysics and the marriage between biology, physics, and chemistry is happening right here in the ATP synthase molecule. And there's this basic relationship that it takes about three protons to make one molecule of ATP. That's the basic stoichiometry of the system. Okay, so kind of in text form, the C subunits, so kind of using the same terminology that we use to describe the flagellar motor, these C proteins would be called the rotor because they are rotating, whereas the F1 subunit is a stator. And the stator down here is connected to by an offset axle, which is what I would call a camshaft, meaning it's not a perfectly round shaft, and it causes this system here to bulge out. So in other words, this whole subunit here is not rotating. It is the axle that's rotating, but yeah, they are moving. But they're still considered a stator. You need something that doesn't rotate in order to capture the energy and capture the energy of that torque. And so, hey, it's another incredible biomotor. where sort of like we learned how to build the internal combustion engine a lot later than this is already out in nature, out in creation. Okay, so 9:15, I think I can, let me look on my jump drive. I loaded this, I hope, the movie, allowed block content. Okay, everybody, I have allowed block content, I hope this is cool. Do you want to allow the following program to make changes to this computer? So I've got to take responsibility for these changes. I want to set this password. No, you chumps. This is an administrator. See, I'm not an administrator. I'm just a lowly faculty member. Okay.
The concentration gradient of protons across a membrane drives the synthesis of ATP by a large complex in the membrane called ATP synthase. ATP synthase, also called the F1-FO ATP synthase, has two main parts, F1, which protrudes into the cytoplasm, and FO, which sits in the membrane. FO consists of 12 identical subunits designated C. A central stalk, called the gamma subunit, extends from FO through F1. Surrounding the gamma subunit in F1 are three pairs of alpha and beta subunits, which service the sites of ATP synthesis. Other subunits, designated A and D, connect the F1 knob to the membrane. The proton gradient across the membrane drives FO to rotate. Protons enter through a channel in subunit A and move into the C subunits, causing FO to rotate in 120-degree steps around the axle.
Isn't that cool?
Protons are released from the C subunits into the cytoplasm. Note that as FO rotates, the axle also rotates, but F1 does not.
Yeah, that's the
F1
status.
is held in place by its connection to the membrane through the A and D subunits. However, the axle turning inside F1 causes F1 to change conformation, catalyzing the formation of ATP from ATP, an inorganic phosphate. An ATP molecule is produced for every 3 protons that cross the membrane through the ATP synthase channel. This function, in which a proton potential powers ATP production, is highly conserved. ATP synthase complexes with the same general structure can be found in the cell membranes of bacteria, in the inner membranes of mitochondria, and in the thylakoid membranes of chloroplasts.
Okay, so that's a pretty helpful figure, don't you think? Helpful movie, showing you the moving parts and then how the physics is transmogrified back into the chemistry with the phosphorylation of ADP. So we'll look at another movie on the electron transport system and then finish up this chapter on Friday.